Hi, Jamie Batts, your instructor for Bio 221. This is going to be part 4 of Chapter 26, and I want to diagram out for you real quick the countercurrent multiplier for the loop of Henle so you can understand how that works. I talked about it before, and I will post a video in our discussion page of it, but just so you understand how it works, again, I think drawing it out always helps, because you get like a bigger perspective and it works that other side of your brain. But you have your thin side and your thick side, right? And remember on the thin, and again, there's thick parts and thin parts to both ascending and descending, so this is a paraphrase. But for the most part, you have the descending is thin and the ascending is thick, for the most part. So on the thin side, this is where water is going to be pulled out. Now, again, this is the paratubular fluid in here, and this is the tubular fluid. If we think about the way this mechanism is initiated, as the filtrate flows through here, it has like a normal osmotic concentration. Right? And your book gives you specific numbers. You don't have to know specific numbers. But once it gets over this side, some of these solutes are going to start being pumped into this paratubular space. Ooh, you can't see my dots. They're just dots. It's not that big of a deal. So, solutes are going to start filling this space. Solutes are going across. And what solutes am I talking about here? I'm talking about sodium. I'm talking about potassium, calcium, a little bit of magnesium. So those are the guys that are coming across. And what that does is that's going to change the osmolarity of this paratubular space. And that's going to pull water from the descending side. And as the water comes in and dilutes, more solutes are going to be pumped out. And as more solutes are pumped out, then more water is going to be pumped out. Right? It's a countercurrent multiplier. It's using a mechanism that's going to multiply on itself, because what's happening on this side is going to affect what's happening on this side. Kind of a cool thing. Countercurrent multiplier. So, just wanted to make sure I hit that on the head a little bit for you. Let's talk about what's happening in some specifics here. And you have a little diagram there on page 961 of what's happening in the PCT. You can tell it's the PCT by looking at it because of the microvilli. Also, the little inset is showing you that it's kind of looking at a zoomed in view of the PCT. So if we actually look at what's happening in there, you have a SymPort system set up. The SymPort is using sodium and chloride, or sodium and glucose, or sodium and amino acids. So either way, sodium's coming back in from the filtrate, in through the paratubular cell, into the paratubular fluid, and eventually back into the blood. And so there's also osmosis that's going to occur, right? Just water is automatically going to follow salt. Water follows, the solutes. So because the sodium and amino acids and sodium and glucose and sodium and chloride are being pumped across, water is going to follow as well. On the paratubular side of this process, the outside of the tube, you have some active transport going on with the sodium-potassium pump. You have a SymPort going on, potassium and chloride both being SymPorted out together. Glucose, amino acids, and potassium are also being exported through facilitated diffusion, and water is leaving in via osmosis. So lots of action going on there. What do you need to know? You need to understand how it works. You should understand what is being SymPorted together in the PCT. You should understand the major ions that are being absorbed in the PCT, being reabsorbed in the PCT, I should say. So then we get to our loop of Henle, or our Nephron loop. I learned it back in my day, it was called the Loop of Henle, and now they've changed everything just to be named after the structure. So it is the Nephron Loop now. But you have the descending thin limb, which is where the majority of the water is reabsorbed, just like I just drew for you, and then the ascending limb, which is mostly thick, which is where the solutes are going to be reabsorbed into the paratubular fluid. And remember that paratubular fluid is webbed with paratubular capillaries reabsorbing all that stuff back into the blood. And that's what you see on 962, 963, exactly what's happening with the thick and thin segments there. It's the same thing I just drew out for you. It's just maybe a little bit more detail. On page 964, you see a little chart there with some substances and how they're transported across. You don't need to know that. I may give you an example of some of those on the exam or on a quiz, and say, penicillin, which is transported via active transport. I would put it into context, but I wouldn't have you know that off the top of your head. That's more for a real clinical use, not for our intensive purposes. The next little section, looking closer into how hydrogen ions and potassium ions are secreted and absorbed. This is super important to understand. I'm trying to see if I have a good diagram explaining this. Not really. No, I don't. But I can give you a big picture of everything. And that is to say that, let me erase my little countercurrent multiplier here with my blanket. What you're looking at in this picture is the PCT and the DCT on page 965. So it's showing you different stops along the tour of the nephron loop. And you're looking at how hydrogen ions, sodium ions, and potassium ions are exchanged and how that helps to balance our overall pH. So just paraphrase some things real quick. Remember, a lot of it comes back to hormones and the presence of hormones. In the presence of aldosterone, if we go back and we think about what aldosterone does, aldosterone is it comes from your pituitary, and it's meant to cause your kidneys to reabsorb sodium, right? In reabsorbing sodium, your body is going to get rid of potassium, right? Sodium and potassium are like the antithesis of each other. They're always moving opposite of each other in the body. So if your body's reabsorbing sodium, it's getting rid of potassium. So if you have aldosterone or rising levels of aldosterone, what your body's going to do is it's going to increase its sodium, and by increasing sodium, it's also increasing H2O, right? Water follows salt. So this is a mechanism that's put into place to build our blood volume and blood pressure, right? Basics of chapter 21 or whatever it was. Well, there's a pH effect here as well, because when you increase aldosterone, not only are you increasing sodium and increasing water, but you are, because sodium and potassium are opposite each other, you are decreasing potassium. These are all ions. I think potassium's +2. Right. No, that doesn't. It's not labeled in your book. Potassium. So you're decreasing potassium. When you decrease potassium, you're also decreasing hydrogen ions. Right? And hydrogen ions are related to your pH, right? The lower your pH, the higher your hydrogen ion concentration is. So if you're decreasing your hydrogen ions, you are increasing your pH. This is all linked in chapter 27, the next chapter, when we look at acid, base, and fluid balance. This is just a little snapshot of it. So just get a little taste of acid-base balance here in the kidneys as we're understanding it. So you can see in, let's look at A, for instance, on page 965. What's happening in the PCT? So it says the hydrogen ions are secreted into the filtrate by this antiport mechanism, right? And what you see there is the hydrogen ions coming in and carbon dioxide is leaving, right? Hydrogen and sodium are moving opposite of each other, which makes sense here, right? Hydrogen and sodium are moving opposite of each other here in this little equation. So as the hydrogen is coming into the filtrate, as our body's trying to get rid of hydrogen ions, it's going to reabsorb the sodium ions. And that hydrogen came from our bicarbonate buffer system. If we look at the origins of all of this, it all starts with carbon dioxide. The carbon dioxide, which again is floating around in the blood, 70% of it is converted into bicarbonate in that sodium hydroxide bicarbonate buffer system. Remember, carbon dioxide and water combine from carbonic anhydrase to make carbonic acid, H2CO3, and this carbonic acid dissociates, and it dissociates into a hydrogen ion and into a bicarbonate ion. We mentioned briefly something called the chloride shift, and what that chloride shift is it's an exchange between bicarbonate and chloride, right? As that bicarbonate is excreted into the blood, a chloride is then reabsorbed into the capillary or into the cell, and the hydrogen is exchanged for a sodium, as we see in that antiport system there in letter A. So really what we have going on is a sodium bicarbonate system and a hydrochloric acid system. If you know anything about basic chemistry, you know that this is a base, a strong base, and this is a strong acid, HCl and NaHCO3. And so they're always going to kind of move opposite of each other. And if you look at that equation, you look at what's going on in that picture there, just focus on letter A, you see that happening. You see sodium and hydrogen being exchanged for each other. That hydrogen came from here. It came from the bicarbonate buffer system. It's being excreted into the tubular fluid, which means your body's trying to get rid of it. It's putting it into the urine. It can't just take the hydrogen by itself. It has to exchange something. It has to equal things out, and that's what the sodium is doing. The sodium is being exchanged for that. Where is the sodium going? Well, the sodium is then going to go back into the interstitial fluid in exchange for potassium. So again, we come back to this idea of sodium and potassium moving in opposite directions. The carbon dioxide comes in from the blood. The carbonic anhydrase within the tubular cell does this reaction. Our hydrogen can be excreted in exchange for a sodium and a potassium, which comes in to the cell because of the sodium-potassium pump. So that's just a paraphrase. Basically, the same thing is happening in the distal convoluted tubule. Here, we're seeing more action with the potassium, though, because our PCT doesn't have the proper channels to get rid of potassium at that point. The DCT does, and it's all about channels and ion pumps at this level of cellular physiology. So the potassium can leave and go into the filtrate at this point in time in the DCT. It can follow the hydrogen as it needs to and be exchanged for the sodium, as I talked about. So that's kind of what's going on there in terms of the big picture. Let me just check, make sure I'm hitting all the big guys. Buffer systems we'll talk about in the next chapter. Okay, page 967 shows you numbers again. Don't be concerned with the numbers. Look at the big picture. When the filtrate comes into the PCT, we're looking at an osmolarity around 300. By the time we get down to the loop of Henle, we're in the 600s. Then we're in the 1200s. So we're super concentrating this. Then we get into right by the collecting ducts, and letter A. This is all through, just making sure we don't have the differences here. So, I'm sorry, I just lost my train of thought. You can see the actual numbers comparing the blood and the loop of Henle in these two diagrams if you look at letter A. I almost wish they would have interlocked them so you could compare them right next to each other, kind of like what they did in C and D. But either way, you can tell the osmolarity changes as the filtrate moves through this system. This is all slightly different in different types of nephrons, which I think I will, since I'm over 15 minutes, I think I'll do that on the next video, part 5, which will probably be the last video. Should be. Okay, bye.